I hope I am audible. Can anyone? Okay, so I'll quickly share my screen and we'll start with our session. I hope my slides are visible.
No, it's not visible at all.
Sorry,
No, it's not visible.
slides are not visible. Okay, so
No, I think you can put it in slide share mode.
okay,
Otherwise, they are visible. Yeah, now it is visible.
okay. Thank you.
No, still it's not showing to me.
Still, it is. Can you leave the session and rejoin the session? There might be some issues from your end because other might see it and you cannot.
Okay, ma'am. Okay, ma'am.
Yeah.
Okay, ma'am.
I hope it is visible now.
Ma'am, do we need to do simultaneously
Sorry.
whatever you like? Should we need to do the simultaneously whatever you are showing?
No, it's not like that. There will be questions and there will be like it would be more of an interactive session than the practical one.
Okay.
Yeah. So I hope my slides are visible to everyone now. Okay, so we'll start with our session. So I welcome you all to the problem solving session for week 5. Um the topics that today we'll be including are Bowtie 2 tool, mapping read with Bowtie 2 output, SAM and BAM format, and SAM format, the alignment section. So the questions that today we'll be discussing from these lectures. So so I'm again hoping that there will be a questions and you guys need to answer that first and then we'll see how what are the concept behind those questions. So the professor for this course is Professor Riddhiman Dhar. He is professor at IIT Kharagpur in Department of Biotechnology. So I'll quickly, briefly introduce myself as well. So I am Pooja Yadav. I am a PhD scholar and a PMRF fellow in Indian Institute of Technology, Bombay. I am in Department of Biosciences and Bioengineering. I am a PhD fellow in Molecular Virology lab. So we'll start with our session now. So we'll start with our first question. I'll just see. Okay. So in a question, there is reference sequence and a query sequence is given. And what is asked is this SIGAR SCIGAR string for the alignment in the figure is, and there are some options given. Please try to go through the question and try to answer this question. I hope my slides are visible to everyone.
Yeah, ma'am, it is visible, but ma'am, most of the options are wrong. No, it is actually
Okay, so I'll tell you just ignore the first three and then try to answer this. Ignore the first three sequence and then try
okay.
to
Okay.
answer this. Yes.
Ignore the first three. Okay.
Yeah.
See, madam.
Okay. Is there any other answer apart from C? Okay, so the correct answer is C, and we'll look into why the C option is correct. So first we'll look into what is cigar strings. So I'll just go through this reference. So this reference, it leads here. I hope you can see it. I hope you are able to see my screen.
Yes, ma'am. Yes, ma'am. It
Is
is
my
visible.
screen clear or it's not clear?
It is little blur
Okay, I'll
actually.
just connect to my phone because I think the Wi-Fi is not. I hope it's better now. There is no difference, right?
Still, I think it is blur only.
It's blurred only, right? Okay, I'll just take a minute. I think
Yeah,
it's
now it
better
looks good.
now, right?
Yeah.
Okay. So in the last lecture, last previous week lecture, we have seen that we have mapped our reads to the reference. In uh, in this week lecture, now we are aligning our reads to the reference sequence. So this Bowtie 2 is a tool or a software that is used for the alignment of our reads to the reference sequence. So after aligning, we get a file which is a SAM file, sequence alignment map. It is a text-based file that holds the alignment information. It is a file which gives us the information of the alignment of our reads to the reference sequence. So what it carries, it has two sections. It has header section and it has alignment section. In header section, we have few paths like HD, at the rate, SQ, and I'll tell you what are those. So in the second part, we have alignment section, and the total of it, the header and the alignment section, they are basically of 11 options which are mandatory for each SAM file, and there are other options which are additional options to this SAM file. But the 11 options, they will always be present in the SAM file. So in the alignment section, we have few options. So one of the information that is given is cigar, where they shows how the alignment has happened. Are there any indels? Are there any insertion? Are there any deletion present? Or if they are present, how many of the deletion or insertion or how many base pair are actually matched to our reference sequence? All the information is given through this cigar string. So this fields come under the alignment position. So I'll just quickly show how the SAM file look like. So after the alignment, we get this sort of file. So this HD, SQ, PG, it is the header information, and here you see this basically these lines. It shows the reference genome or the this chromosome number, or this is the, here you can see it is the cigar string information. It is 100M. Basically that 100 of the bases has aligned to our reference genome. So like that, this is a SAM file, a part of a SAM file, the initial, the head of the SAM file, where a different information is given, including the head information as well as the alignment information. So in this question, what is asked is, we need to find the cigar string of the query sequence. So here, so if so there are certain alphabets that are related to certain annotations. So M is for alignment match, that our base has aligned to at the correct position to the reference sequence. I stands for insertion, D stands for deletion, N stands for skipped region, S is soft clipping, H is hard clipping, P is padding, equals to sign is for the sequence match, and X sign for the sequence mismatch. So here we need to find the cigar string, and if we ignore these first three, here we can say that two bases are matching correctly to our reference sequence, and then there is a gap for three bases. So 2m 3, the gap denotes here deletion, so here it will go 3 deletion, and then again the 4 bases are correctly matching to our reference sequence. So the correct answer would be 2m 3d and 4m. So I hope this is clear. Is there any doubt? Oh, slides are not clear. Slides are still not clear. Slides are clear now, or they are still blurry? So I hope this
Better, ma'am.
question is clear.
They're better.
If there is any doubt in this question, you guys can ask me. So if this is clear, the correct answer would be C, which is 2m, 3d and 4m. M for mismatches, D for deletion, then again M for the mismatches.
Ma'am,
Okay.
M is for match, right, ma'am?
Yeah, sorry, M is for the match. So I hope this is clear. So if this question is clear, we'll move to our next question. Question number 2. The explanation of flag is given as this is a paired read. It is the first in the pair and its mate is unmapped. What should be the flag value for this explanation? Use the table below to estimate the value. So okay, so we have a read and the flag, the flag, the flag, the explanation of flag is given. So basically, the flag tells you how the reads are mapped to the reference sequence. So by that we give number to each components, whether they are mapped paired correctly or not paired correctly. So based on that there are numbers given to a certain option. So here in this question it is already given what are the correct alignment for this reads. So this read is a paired read. It is the first in the pair and its mate is unmapped. So paired read is, we here it is pair and sequencing is done and there are two reads, read 1 and read 2. So based on that you can try to answer this question.
C, madam.
Okay.
Then B.
Okay. So B and C, we have two answers. So apart from B and C, is there any other answer that might be correct?
I think it's B only, ma'am. B. B is the correct one.
Okay. Yeah, the correct answer is B. So okay, I'll just copy paste it here. Here what is given, the read is paired. So for the paired read we have 1 and the mate, the read 2 is unmapped. For the mate which is unmapped we have 8 and it is also given it's first in the pair. Basically it's the read 1 and for that we have 64. So So the flag would be 1 plus 8 plus 64, that would come to 73. So I hope this question is clear. So the correct answer for this question would be 73, the option B. I hope this question is clear. Can anyone confirm? Okay.
Yes, ma'am.
If you guys have any doubt in any of the question, you guys can interrupt and ask any questions. So, okay, now we'll move forward with our question number 3. Okay. So, the question number 3 states that when does R name shows star value in a SAM file? So, A, if at SQ header line are present. B, if all the reads come from the same template fragment. C, when the query sequence is absent. D, when a read fails to align against the reference sequence. Please try to answer this question.
D, ma'am.
Sorry, can you repeat?
D.
Okay. We have D as one of the answer. Is there any other answer that might be correct? Okay, so D is correct. So R name is basically the name of a reference sequence where the alignment occurs. So basically it can be the chromosome number from where our reads are getting aligned, to which our reads are getting aligned. So if in our file, if we have instead of in place of R name we have a star and not the exact chromosome number or a sequence, and if the star is given, it means that there is no, when it, the read fails to align to the reference sequence. So we don't know, and we don't know any region where the of the reference sequence where the read is aligning. So in this in this a read fails to align to the reference sequence, so we don't get the information about a reference sequence. So when the information about the reference sequence is absent, so we get this star. The file shows the star in place of the exact reference sequence. So it can be any chromosome number. If our reads would have aligned to any chromosome, then it would have shown this chromosome number, the exact chromosome number. But if our reads fail to align to the reference sequence, it gives this star. So if we go to this right, can you see my screen? I hope it is visible. So here you can see we get the information of the chromosome number from here as well. So our reference sequence where our reads are getting aligned is chromosome number 20. But if our reads are not getting aligned to any of the chromosome or any of the sequence, we get star in this, at this place. So I hope this is clear. So the answer would be D, when a read fails to align against the reference sequence. I hope I am clearly audible to you guys. Okay.
Yes, ma'am.
If question number 3 is clear, we'll move to our question number 4. Okay. In zero-based coordinate system, a region between the 10th and the 15th base is inclusive is denoted by. Please go through this question and try to answer this question. Sorry,
D.
D. Okay.
Getting confused between B and D, but I think it's D.
Okay, it's a zero-based coordinate system. So
Yeah.
yeah. So just remember this and try to answer this question. Because in zero-based coordinate system, we start the position that we give a zero to our first base, not one. So do remember it. Kanchan, there might be few extra question or few question that might not be this there in the assignment, but again, they all the questions that are there will help you to do your assignment. So there might be few questions that might be here and there, but not all the questions, hopefully, because I have not seen the assignment yet.
Okay, ma'am.
Yeah.
Thank you.
We have one answer C and one D for this question. Please try to answer this question. Take few minute time, but trying to answer this question. Okay, the correct answer for this question is C. So, okay, we'll look into what is zero-based coordinate system. So, a coordinate system where the first base of a sequence is numbered 0. So this is zero-based coordination system. We have a coordinate system. So we'll look here how this is. We have first zero-based coordinate system, and then we have one-based coordinate system. When our first base is numbered at 0, we call it zero-based coordinate system. But when our first base is numbered as 1, then it is first one-based coordinate system. So what happens in the zero-based coordinate system? So counting starts from 0, making the first base position 0. It is half open interval. So these bracket and this parenthesis, it is the half open interval. So it is not a closed interval, it is a half open interval. It includes the start position, but it excludes the end position. Why it is excluding the end position, we'll look into that, making it easier to handle and calculate in the computational task. So here we are given with 10th and the 15th bases. So in the zero-based coordinate system, the 10 refer to the 11th base, because our first base is numbered at 0. So this 10 would refer to 11th base, and this 15 it would refer to 16th base. So that is why it is not a closed interval, but it is a half open interval. So this bracket 10 comma 15 parentheses, it includes this 10, 11, 12, 13 and 14, which corresponds to 11, 12, 13, 14 and 15 bases in a first base system. I hope you guys are getting this. This annotation is actually important. So why this annotation is, or this notation is used? It is a common because it allows for a easy calculation of the length of the interval. If we simply subtract the end position, base position, to the first position, we know how many bases are there. But if we do the same for the first based coordinate system, we won't get the correct answer. Suppose here we needed to add one or subtract one basis to get the actual answer. So I hope this is clear. So the correct answer would be C for this question.
Ma'am, when you use the word inclusive,
Okay.
is there any other thing like exclusive, some kind of thing like?
In what context you are saying? Can you like remind me?
Because the question says inclusive.
Okay.
Inclusive
Okay.
denotes, yeah. Anything like
No,
opposite
it's
kind
not
of-
exclusive and all. There is no notation of exclusive and all. It is just they are including these position.
Okay, ma'am.
Right. So I hope this question is clear. So if this is clear, we'll move to our next question. Question 5th. Okay. So if the probability of being a map position wrong is 0.7, what will be the map Q value of the read? So map Q is the mapping quality. So how can we calculate the mapping quality if we are given with the probability of being mapped wrong, position wrong? So please try to solve this question by your own first.
Ma'am, A is the answer, but I used calculator because
It's
I
all
think
right. Okay, now we have a one answer, which is A. Okay, is there any other answer apart from A which might be true or correct? Okay, we'll see how to calculate map Q value. So the map Q value is calculated by the formula minus 10 log 10. The probability of mapping position is wrong. So we are given with this value. We are given with the probability of mapping position is wrong, which is 0.7. And if we substitute this value to this formula, we get the map Q value of 1.549. So this quality is score, or the quality value is again the same concept that we initially read when we were sequencing and we were finding that exact base is coming to like this position or this read only. So this is exactly how it was also calculated. So we get this formula by only substituting the value of the probability of mapping position is wrong. We get 1.4549. It's again a very straightforward question.
Madam,
Yes.
Madam, actually, if such question comes
Okay.
without calculator, I don't know how to calculate log 0.710. So that,
Okay, you guys can use the calculator only. But don't use the phone calculator.
okay.
Yeah.
I'm asking in context of exam, I think.
Exam, they will provide guidelines actually, so
Okay.
you will know whether to use calculator or not. Most likely you will use the calculator because there are many numerical questions, so for that. But again, zero point 0.7 log value is like common, we know at least. नहीं 0.7 का तो नहीं पता, right? But it's all right. I think you guys can use the calculator but not the phone calculator. You cannot carry your phone. So
Thank you.
okay, so if it is clear, so we'll move to our next question, which is, so the correct answer for question number 5 is A, which is 1.549. So we'll move to our next question. Okay, so what is hard clipping? A, bases are not aligned and not present in the read sequence in the SAM file. B, bases are not aligned but present in the read sequence in the SAM file. C, bases are aligned and present in the read file in the SAM file. D, when the sequence is completely matched. So please try to answer this question. It is again one of the value of the cigar string, so please try to remember the values of the cigar string and try to answer this question. We have one answer, C option.
I think B, madam.
Okay, so we have two options now, B and C. Okay, is there any other option apart from B and C that might be correct? Okay, so we'll look into what is hard clipping. So hard clipping is when the reads are not aligned and the clipped nucleotides are not present in the read. So if the clipped nucleotides are not present in the read, it basically won't align. So the correct answer would be A. The bases are not aligned and not present in the read sequence in the SAM file. I hope this is clear. Okay, if it is clear, we'll move to our next question. Question number 7. Question number 7 state, in a SAM file at the header position at HTVN 1.3 is written, what is the significance of this line? A, this header records the reference sequence version with the tag VN. B, it records the SAM version with the tag VN. C, it indicates the sort order. D, it shows the version of the mapping program. Please try to answer this question. So it is the one of the option which is present in the header, this portion of the SAM file, which is HDVN. And if VN, so what this VN 1.3 it denotes in the SAM file.
I think it's A only, madam.
Okay, so we have one option which is A. Is there any other option which might be correct for this question apart from A? We have B option as well. Okay, so we'll look if so. We have this. Where is it? BN. Okay, so the VN basically shows the SAM version. So it is not the reference sequence version, it is the SAM version. So here you see in the header we have this HD and this VN with the VN tag, and the VN tag value here it is given 1.0. It is showing the version of the SAM file. It is not showing the version of the reference, because reference is the sequence. It is not a file or something. It's a sequence. But for SAM file we have the version which is in this question it is given 1.3. So it records the SAM version with the tag VN. I hope this question is clear. Okay, if it is clear, we'll move to our next question. Choose the true statement about mapping among the following. Please go through the options and try to answer this question. Take a few minutes on this question and try to answer this question.
A, D, and F.
Okay. We have one correct option. This includes A, D, and F options. We have other option A, C, and F. Is there another option that might be correct for this question? I hope my screen is visible. Is my screen visible? Are my slides visible?
No, ma'am.
Okay, I'll just share the screen. I hope my slides are visible now. Okay, so we will look what option might be correct for this question. Okay, so basically what is concordant mapping and what is discordant mapping? So it is basically for the paired end read sequencing only. It is not for single end sequencing. So in paired end sequencing we have two reads, read 1 and read 2, and then we have a reference sequence where we are aligning those reads. So what we are expecting during the alignment, that are read 1 and read 2 which is coming from the same fragment. Remember, these reads are coming from the same fragment, and it will go and align to that specific DNA fragment. And so if read 1 and read 2 is aligning to the same DNA fragment, there might be some overlapping that can be possible in the read 1 and read 2 alignment. But what happens in the discordant mapping? So in discordant mapping, so read basically our reads are not aligning at the exact position of the reference. One of the read might not be aligning to the same DNA fragment, or it is aligning to a reference or a DNA fragment coming from a different chromosome itself. So there might be some recombination or some structural variant that might be happening during during certain criteria. So let's suppose if we are studying like a genome sequence, where a certain mutation has happened in the genome and a certain structural variant mutation has happened. So recombination of the genome has happened in the genome, and we then sequenced our this DNA from that particular organism, and then we have the two reads. But when we map those reads to those using the same reference sequence, our one of the read is not aligning to the same position because there might have been recombination happened in the chromosome. So one of the reads, so one chromosome has only this part of the uh, uh, this part of the reference sequence, and the other chromosome might have this part of the chromosome. So here the read won't be aligning at the same DNA fragment. So there is no possibility of the overlapping between these read 1 and 2 in the case of discordant mapping, but it is possible in the concordant mapping. So the correct answer would be A. In concordant mapping, there is a possibility of overlap between read sequence. D. Concordant and discordant mapping, it is only applicable for pair and reads. And F. In discordant mapping, there is no possibility of overlap between read sequence. So I hope this concept is clear. What is the difference between concordant mapping and discordant mapping? So when we study the mutational studies, so there might be different mutation has happened. Some of the mutation is generating the structural variation in the chromosome. So this discordant mapping is mainly useful where we can find these structural variants during our mapping or mapping this during while we map our reads to our chromosome. So I hope this is clear. So the correct answer would be A, D and F. this question is clear, we'll move to our next question. Question number 9. Two mates are mapped uniquely and their starting and ending position are the same, but they are mapped in a different two different chromosomes. What kind of mapping is this? A concordant, B discordant, C unmapped, D none of the above. So it's we have just um, studied the concept for this. Please try to answer this question.
B, madam.
Yeah, the correct answer for this question would be B, discordant. So if it is clear, we'll move to our next question. If the TLN section in SAM file for a read sequence has a lower value than the actual length of the read sequences, what of the following statement is true? A. There is a snip in a read sequence. B. Insertion in read. D. Deletion in read. D. Error in mapping. Please try to answer this question. So we have one option B.
C, madam.
Okay, so we have another option C. We have two options B and C. So is there any other option that might be correct apart from B and C? Okay, so we will look into it. So what is TLN? It's an observed template length of fragment length. So how do we calculate TLN? So TLN is basically the read 1, position of the read 1 and position of the read 2 plus the length of the read 2 if the upstream is read 1. So I will look into this concept later on. So in this question what is asked is if the TLN section in a SAM file of a read sequence has a lower value than the actual length of the read sequence. So this. So here the TLN, so if the wild type TL read sequence is coming to be smaller than the actual length of the read sequence. So it shows in TLN it is showing the read sequence of the wild type, or we can say it's a wild type read sequence, but what is the actual read sequence is slightly longer than what is observed read sequence. So if it is if the actual is longer than the observed and or the observed is lower than the actual, so there might have been insertion in the read. That is why, that is only it is possible that the actual is longer and the or a higher, or the observed one is lower. I hope this is clear. So we have one observed TLN and one is actual TLN. And if observed TLN is lower than the actual TLN, then there might have been insertion in the read sequence that has happened.
Ma'am, actually it is not clear for me.
It is not clear. Okay.
Like when there is a insertion, it should increase the length of the total read, right?
Yeah, it will increase the actual TLN length. But in the question it is given we have a lower observed TLN. So if we have lower observed TLN and then the actual TLN, so there is actually insertion happen in the read. That is why it is actual. But so in this during the mapping we use the actual read length, but the T length, it is given by the observed one. I hope it is clear. So in this, okay, I'll show you. So where is the T length? Okay. Mm. Okay. So in the question, what is given? T length section in a SAM file for a read sequence has a lower value than the actual length. So in actual reads that are present in the file is actually is of the higher value than what we are observing during in the SAM file. Initially, what we have thought that since these are the wild type reads, so the software has picked the wild type reads and it has given the T length for these reads, but the actual reads that we are giving, that we have sequenced, is of the higher T length value. I hope it is not confusing. If it is confusing, I'll explain this in the next class maybe, or maybe in the next question you will get to know how it is done. So here we have the actual T length value is higher than the observed one, which is present in the SAM file. So in question it is given opposite, that T length section in a SAM file for a read sequence has a lower value than the actual read sequence, actual length of the read sequence. So there might have been insertion in the read sequence that has happened. That is why the T length which is given in the SAM file is lower than the actual length of the. So just think it opposite. Just so the correct answer would be B, insertion in read. I hope this is clear, or this might be clear with coming questions. And if it is not clear, I'll explain in the next class properly. Okay, so we'll move to our next question, which is question 11. What does the bow tie to build command do in the read mapping process? So please go through this question, take a few minutes and try to answer this question.
Does it has multiple
Yes,
answers?
yes, it can have multiple answers.
A and B, ma'am.
Okay. So we have one option which is A and B. Is there any other option that might be correct apart from A and B? Okay, so we'll look into the concept. So this command bowtie2 build is used for building the genome index, the reference sequence genome index. The command for this is bowtie2 build. Option we might we can give a multiple option based on our requirement, but the main this is we need to give our reference sequence which needs to be indexed, and this the base name of the index file to write. So what this bowtie 2 build do is it generate an index from the reference sequence, which bowtie 2 then uses to quickly map the sequencing read to the genome. Then after this, it gives the B a file with the extension .b2t extension. Then it is used by bowtie 2 during the alignment process. First, we need to do the indexing of our reference sequence or the reference file. Then it gives the this .b2t extension file, which then giving the bowtie 2 command, this file is used for alignment of our read sequences. So first we need to do the indexing of our reference sequencing, and then we get the file which is readable to this bowtie 2, which is having .b2t extension, and then we give the bowtie 2 command, and by giving the bowtie 2 command, the alignment, we get the alignment of the read sequence to our reference sequence. So there are two processes in this bowtie 2. First, we give bowtie 2 build command. It is again for the indexing. Then we give the bowtie command where the alignment actually happens. So the correct answer would be A and B. Bowtie 2 builds create an index for the reference genome, enabling fast and efficient alignment of the sequencing read. Files with .b2 extension are generated for index files. So after indexing, we get .b2 bt2 file, and this file is used for the alignment by the command bowtie. I hope it is clear. So if it is clear, we'll move to our next question. Question number 12. If you know the position of mate 1 and mate 2, how do you calculate the fragment length, which is T-len? Mate 1 is located upstream of mate 2. Please go through this question, take a few minutes and try to answer this question.
B, madam.
Okay, we have one option which is B. So do remember that mate 2 is located upstream of mate 1 is located upstream of mate 2. Remember this and try to answer this question. Upstream basically is mate 2 is aligning to 5' of our DNA fragment, while the mate 2 is aligning to 3' end of our DNA fragment. So mate 1 is located upstream, like near the 5' end of the DNA fragment. We have two options, B and D. apart from B and D, is there any other options that might be correct? So we will look into it. So what is given in the question is make 1 is located upstream of make 2. Basically read 1 is located upstream of the read 2. So here we have a reference sequence. We have read 1 which is at the position of 5' end of our reference or the DNA fragment, and read 2 is at the 3' end of our DNA fragment. So to calculate the T-length, so we first find the distance between the position of the mate 1 and mate 2, the starting position of the mate 1 and the mate 2. The starting position is basically from the left side. So for the read 1 or mate 1, we have the starting position from here, and for the read 2, we have the starting position starting from here. So if we have the position between the read 1 and read 2, which is covering this length of our DNA fragment. So to completely calculate the entire T length, we need to add the length of our mate 2 as well. So first we need to find the position distance between the position starting position of read 1 and read 2, then we add the actual length of the read 2 to get the entire T length. I hope it is clear. So by this the correct answer would be D, position of mate 2 to position of mate 1 plus read length of mate 2. I hope this question is clear. Can anyone confirm if it is clear till now?
We were calculating like read 2 minus read 1 plus 1. No,
No, it's not read 2 minus. It's position of mate 2 to position of mate 1.
Actually, mate 2, how you told that is starting from this end of the read 1? Because it should start from the right extreme, right? If your read 1 is coming from the left extreme.
Oh, right. You are actually right. I got confused with this question. So it is minus only, right? And it is starting from the right end of the. Sorry. I got little confused. So if by this, if we are starting with this position of the read 1 minus position of the read 2, and then we calculate the read 2 length, then we get entire T length. So sorry about that, I got little confused with this question as well. Thank you so much for pointing it out. I hope it is clear, the confusion is clear. Can anyone reply if this is clear? Okay, if it is clear, we'll move to our next question, which is question 13. P and X values in the cigar sequence means A. Soft clipping and hard clipping, B. Mismatch and soft clipping, C. Padding and mismatch, D. Hard clipping and padding. So we have seen multiple options for multiple values for the cigar sequence. This question is based on that. We have P and X, and this question is asking what P and X means in reference to the cigar sequence.
Is C, madam.
Okay. We have one option C and one option D. Is there any other option that might be correct apart from B and D and C? Okay, so the correct option would be this C. So P stands for padding and X stand for sequence mismatch. The correct option would be C, padding and mismatch. So I hope it is clear. Okay, then we'll move to our next question. What does YT colon Z colon UP at the end of a line of a SAM file means for an aligned read sequence? A. The read was a part of a pair and the pair aligned discordantly. B. The read was a part of a pair and the pair aligned concordantly. C. The read is not part of a pair. D. The read was a part of a pair but the pair failed to align either concordantly or discordantly. Please go through this question. Take a few minutes and try to answer this question. Okay, we have one option which is D.
A, ma'am.
Okay, so we have another option which is A and D. So apart from A and D, is there any other answer that might be correct? So basically, again, you cannot remember all these, like all the field values. So it's sort of a memorizing question only. So what is given is YTZUP. So it stands for the read was part of a pair, but the pair failed to align either concordantly or discordantly. So the correct option would be D. You can refer to like any reference. You don't need to memorize it. You can refer to any of the this. But there might be question in the exam, so you need to. I mean, there will be certain reference that might be given for the question, because of course you cannot memorize all the fields. So I hope this is clear. So if it is clear, we'll move to our next question. Okay. So certain information is given. So let's consider two paired and reads, read 1 and read 2. Read 1 has a flag value of 99. Now based on the given information, answer question 15 to 17. Okay, the question number 15 is what will be the flag value of read 2 if mapped properly. So here it is, so the table, I have pasted a table here, so you can refer this table and try to uh, calculate the flag value of a read to if it is mapped properly. Please take a few minutes and try to answer this question.
One 147, ma'am.
Okay. So we have one answer, which is 147. So basically, these are all the fields, uh, how you can analyze your alignment data. So certain fields are given, so you can properly go through it and analyze your alignment, whether it has aligned correctly, or if it is aligned correctly, where it has been aligned, or what is the score of the alignment. So you can rely on your data which you got more accurately, and or whether if the alignment is proper or not. You can get all these by these field value which is present in the SAM file.
Ma'am, you came out
Yeah,
of
yeah.
slides.
Just okay. I hope my slides are again visible. Yeah. So okay, what is given in this question? We need to find the flag value for read to, and it is mapped properly. So what sort of information is given already, that it is a pair and read.
Paired.
Yes, and it is also mapped properly. So based on that, we have, it is read paired, it is a pair and read, so one for pair and read, and it is mapped properly, read map is in proper pair, then we have two. Read reverse strand, so we know that it is a read 2, so it is the read align in the reverse direction, and we know that read 1 is upstream of the read 2, so it is 16 for this one. And we again know it is read 2 second in pair, so that to 128 for second in pair. So if we add all the values, we get the flag value for the read 2, which is 147. So the correct answer would be 147. So I hope this question is clear. If this is clear, we'll move to our next question, question number 16. If the pause value are 1000 and 1200 respectively, is it the concordant mapping? A option is yes, B option is no. So So we know what are the this is pair and sequencing. We are given with read 1 and read 2, and we know that read 1 is upstream and read 2 is downstream. And now here in this question, the position of the read 1 and read 2 is given, and we need to uh, by all this information we need to find out whether it is a concordant mapping or it is a discordant mapping. It just key like it is just asked, is it a concordant mapping?
It is
Yeah.
no, madam.
Okay. If no, why?
No.
Okay. So can you explain why it is no?
The difference is more than the T length, right?
How did you calculate T length here?
Sorry. I did a mistake. Yeah.
Yeah, it's okay.
I confused flag value as T length.
Yeah, it's okay. So we have one option, yes. Okay, so we have yes and no. So it's yes, it's a concordant mapping. So what is given? So what are we given with? The flag value for read 1 is 99. So what it says that the read is paired, the read is mapped in a proper pair, and the read is the first in the pair, and the read is mapped to the forward strand, and the read is mapped to the reverse strand. Okay, so for read 1 we know that it is pair and sequencing, and the read is mapped properly, and it has mapped to the forward strand. And from read 2 we are given with the information that it is again a pair and sequencing, and uh, it is mapped properly. And if read 1 is mapped to the forward strand, the read 2 is mapped to the reverse strand. So now we are given with the position of read 1 and read 2, and read 1 position is smaller or the lower than the read 2 position, which is intuitively should be correct, because read 1 is coming initially to the read 2, because it is the upstream to the read 2. So the position of the read 1 should be lower than the position of the read 2. In this question it is given the same, and when we minus position of the read 1 to read 2, it is coming around 200. So which is basically the basis between the read 1 and the read 2. So which we know that 200 base pair read length is possible. It is not very much, I mean it is not too long, it is just the 200 base pair. So we can intuitively say that it is a concordant mapping, not the discordant one, because the position are the read 1 position is lower than the read 2 position, and when we see there is a difference of just 200 base pair between the two. I hope this question is clear. So if there is no doubt, so we'll move to our next question. Question number 17. If both the reads are unmapped, what will be the flag value of read 2? Okay, so I'll copy paste here. Please try to answer this question. Take a few minutes and try to answer this question. We have one option, which is one answer, 133.
141, ma'am.
Okay. We have two options, 133 and 141.
Getting confused between 141 and 137, ma'am.
Okay, so we have three options, 133, 141 and 137, right?
Yeah, ma'am, 137.
Okay. So is there any other answer that might be correct apart from 133, 141 and 137?
The 141 or 137, either of the one.
Okay. So we will look into it. So, okay, just. Okay. Okay. So in this question, what information is given? We need to suppose that reeds are unmapped, both the reeds are unmapped, and then we need to find the flag value for the reed 2. So we know that these reeds are pair end reeds. So one would be for the pair end, and then the reed 2 is unmapped. So this reed was not aligned. The 4 would be for unmapping reed. 8 is for the reed is a part of a pair, but its mate was not aligned. So its mate, which is reed 1, it is also not aligned. And 128 is for the reed is the second in pair, which is reed 2. So if we add all these value, we get 141. So here you were correct. So the correct answer is 141.
Excuse me, ma'am.
Yes.
Ma'am, actually I attended the session with Namrata ma'am as well, and she discussed the answer as 73.
No, the 73. Did you join the class late or
A little late, ma'am.
a little late? Okay, the 73 is for this question number 1, not the question number this one. You might be confusing with the question number 1. Not, sorry, the question number 2.
Okay, ma'am.
We have already discussed it. Yeah. So let's go. So okay. So the correct answer for this question would be 141. So then we'll move to our next question, question number 18. In the context of the sequence alignment using cigar string, what does the padding refer to? A. Adding extra bases to the beginning of the reference sequence. B. Filling in gaps in the read sequence caused by sequencing errors. C. Inserting additional bases to align the read sequence with the reference. D. Identifying how insertion in one or more read affect the alignment amongst the read. Please go through this question and try to answer this question as well. Okay, so we have one option. There might be change in few questions, but I think that the question that we have discussed might help in solving the questions that are present in your assignment. I think these problem solving questions are little modified than the questions that you guys are getting for the assignments. They are not exactly the same questions. I mean, many questions are same, but there are few additional questions and few questions that are not in this solving session as well. So we have two option for this question, C and D. Is there any other answer that might be correct for this question apart from C and D? Okay, so we will look into the concept. The D option is correct. The padding, it gives us the idea about how inserted sequence in the read affect the alignment against each other. So let's suppose we have this reference sequence, and when we calculate the cigar value for all the reads that are given, so for the read 1, so there is insertion of GA that is happening here. So we would get the cigar value for this is 8M to I and 9M. I stands for insertion, M stands for match. Then we have initial 8 base pair match. Then we have 2 base pair insertion, and then we have a 9 base pair basis of match with the reference sequence. And here we are seeing that insert is happened in a read 1. So what padding is used for, it gives us an idea how insertion in one of the read affects the alignment of other reads. So in read 2, here this insertion of only one base has happened, and there is, this, you can see a star here. So what it denotes is there is initially 8 base pair match, then 1 base pair padding, then 1 with it is with respect to the read 1. The padding is respect with the read 1, where we saw the 2 insertion, the initial 2 insertion that has happened. Then there is 1 base pair insertion, then again there are 9 matches. And when we look into read 3, it is same as that of the reference sequence, but with when we compare it to read 1, there is 8 matches, 2 padding, and 9 again the 9 matches. So at padding, it gives us an idea about how inserted sequence in one of the reads, how it impacts the alignment of the other reads. So this is given by the padding value, or the padding of the sequence. So the correct option would be D, identifying how insertion in one or more reads affect alignment amongst the reads. I hope this question is clear. Okay, so this was our last question for today's session. So is there any doubt in any question, or like a general doubt in with, because we are done with our today's session. I guess if there are no doubt, we can end our session here and we will meet again the next week with our next problem solving session. So, thank you all for joining. So, we'll meet for the next session.